The next presentation will be held by Dr. Nunzio Denora. Dr. Denora, after its PhD in pharmaceutical technology at the University of Palermo and the postdoc at the University of Kansas, is working at the Faculty of Pharmacy of the University of Bari. His research are focused on new drug conjugate useful to overcome the blood-brain barrier, the developing of nanocarrier, and the role of the benzodiazepine receptor. The presentation of today is Dopamine and L-Dopa Prodrugs, new brain delivery system. Please, Dr. Denora.
Thank you. Good afternoon, everybody. So let me spend just a few seconds on the current therapy on Parkinson's disease. As is well known, the L-Dopa is the only therapy used for the treatment of Parkinson's disease, but the long-term use of L-Dopa is associated with abnormal involuntary movements known as dyskinesias. And furthermore, the L-Dopa is metabolized to dopamine in peripheral tissues, giving a lot of side effects like nausea, vomiting, and hypotension. So different approaches, new approaches have been done in order to provide a continuous delivery of L-Dopa or dopamine to the striatum. And they are all summarized in this nice table. In general, I can say that there are two non-invasive approaches to deliver a drug or drugs to the brain. The first one is a pharmaceutical technology strategies that is based on nanosystems, and the other one is a medicinal chemistry-based strategies that is based on the chemical modification of drugs in order to increase their lipophilicity or to obtain a co-drug or a prodrug. So we have different prodrugs about dopamine and L-Dopa, and a good example are presented in this slide. As you can see, Boder presented the chemical delivery system of dopamine, and so it's based on the pyridinium dihydropyridine redox carrier. Another good example is the tripeptide prodrug that is based that is targeting the intestinal peptide transporter, and another good example was proposed by Bovine et al. And is a glycosyl dopamine or L-Dopa derivatives. And in the end, another one can be the dimeric prodrug proposed by Di Stefano, where two identical drugs are linked together through a very short spacer. We have also recently reported a new L-Dopa and dopamine prodrugs containing 2-phenylimidazopyridine moieties. In particular, the general structure is reported in this slide, and these products have been prepared in order to take advantage of two features concerning the imidazopyridine compounds. The first is the high affinity and selectivity that is shown for the GABAergic receptor. That is shown from most of the imidazopyridine compounds, such as the anxiolytic agent zolpidem. And the second is based on the high lipophilic nature of the imidazopyridine moieties that can serve to carry the dopamine or L-Dopa into the brain. So after the prodrug, another good approach to deliver dopamine and L-Dopa to the brain is by using the co-drug approach. This strategy offers an improved delivery when it is desirable to have at least two drugs, two different synergistic drugs that are linked together, and they are able to reach at the same time the same site of action. So there are two examples in this slide. One was proposed by Pinnen et al., and where dopamine and L-Dopa were linked with the antioxidant glutathione. And another example was the L-Dopa linked with the catecholmethyltransferase inhibitor entacapone. So we are now working on Prodrugs. In particular, we are working on new dopamine and L-Dopa Prodrugs, the so-called PCPDOP compounds. And the general structure is in this slide. As you can see, dopamine and L-Dopa are linked through a very short spacer to imidazopyridine compounds. In particular, the imidazopyridine compounds can be able to bind with high affinity and selectivity two kinds of receptor. The central type benzodiazepine receptor, that is a GABAergic receptor, and the other one is the peripheral type benzodiazepine receptor. It has been suggested that drugs like the zolpidem and imidazopyridine compounds that are able to enhance the GABA-aminobutyric acid transmission could be useful in the treatment of Parkinson's disease. In fact, the GABA-A receptors are able to modulate the activity of the mesocortical and the mesolimbic dopaminergic neurons. In the other hand, there are several evidences several findings confirmed that there is a correlation between the widespread microglial activation and the pathological process in Parkinson's disease. And I should remember that the activated microglia is characterized by a very high expression, an overexpression of the peripheral type benzodiazepine receptor. So we have recently reported a series of a potent and selective PBR ligands and as well GABAergic agonists, and we have selected two molecules, in particular the CB217 that is a GABAergic agonist and the CB185 that is a PBR ligand. So the idea was to link to these two ligands the dopamine through a very simple synthesis. In particular, is a one-pot synthesis where the ligand were solubilized in anhydrous THF at a temperature of 50 degrees in the presence of bis-paranitrophenyl carbonate. So to give the intermediate that in presence of dopamine or L-Dopa ethyl ester is giving the PCPDOP compounds. We to estimate the ability of these compounds, of these Prodrugs, to permeate the blood-brain barrier, we have first evaluated their log BB values that were evaluated according to the Clark's model. And so the log BB was evaluated by the following equation, where the PCI is the polar surface area and was evaluated by using the EARTH method, and the C log P is the calculated octanol water partition coefficient. So, as you can see from the data that are presented in this table, the PCPDOP actually, the first one is the PCPDOP 11, and the second one is the PCPDOP 12. They are Prodrugs of dopamine. They are characterized by log BB values that are less than -1. It means that these Prodrugs should be able to cross readily the blood-brain barrier. But the PCPDOP 13 and 14, they are L-Dopa Prodrugs. They are characterized by log BB values that are less than -1. So they were not able, they should not be able to cross the blood-brain barrier. At the same time, we have also evaluated the hydrolysis rates of these Prodrugs in phosphate buffer, pH 7.4, and 50% diluted human serum. And the results are also summarized in this table. As you can see, the dopamine Prodrugs are stable in phosphate buffer, moderate stable in phosphate buffer, but they undergone faster cleavage in diluted human serum. The L-Dopa Prodrugs were found to be very unstable in the phosphate buffer. So next step was to evaluate the affinity for the CBR receptor and PBR receptor of the dopamine codrugs. In fact, as you can see from the data, these codrugs are able to bind the receptors, so they are biologically active. Now, to deeper understand if these codrugs are able or not to permeate the blood-brain barrier, we have used a well-established in vitro model. In particular, we have used the bovine brain microvessel endothelial cells by using the side-by-side chamber diffusions. And as you can see from the data, the PCPDOP 11 and 12, they are dopamine codrugs, are able to, should be able to permeate the blood-brain barrier, since their values are very close to that evaluated for the transcellular marker diazepam. Then the other step was to have information about the interaction of these codrugs with the glycoprotein P. In particular, we have used the rhodamine 1,2,3 uptake assay, and the tested compounds were not able to affect the intracellular concentration of rhodamine 1,2,3, so it means that these codrugs are not substrate for the PGP. And of course, we did an in vivo study. In particular, we have used the microdialysis studies, brain microdialysis studies, and as is well known, microdialysis is a well-established method for determining the neurotransmitter concentration in the brains of freely moving rats. So we are using this method to detect the dopamine level in the brain of freely moving rats after intraperitoneal administration. And as you can see, by intraperitoneal administration of the dopamine codrug, PCPDOP 11, there is a dose-dependent increase in the dopamine level, and this result is more evident for the PCPDOP 12, where you can see a dose-dependent increase level in the striatum of freely moving rats, at the same time a controlled release of dopamine. These results are also significant from a statistic point of view. In fact, on the B panel, you can see that there is a statistic. As control, there is the vehicle, just the vehicle, then a doses of 10 micrograms per kilogram, and a dose of 20 micrograms per kilogram. So, in the end, novel codrugs of dopamine have been prepared, so the PCPDOP 11 and PCPDOP 12, but they were found to be adequately stable in buffered solution at pH 7.4, while in diluted human serum they undergone faster cleavage. The computational calculation supported by in vitro transport studies on BBMEC indicated a good brain penetration for the compounds PCPDOP 11 and 12, and then the brain microdialysis showed that these dopamine codrugs induce a dose-dependent increase in dopamine levels in the striatum. And PCPDOP codrugs can be considered as delivery system able to provide a controlled release of dopamine to the striatum. Of course, the next step will be to evaluate these codrugs in a Parkinson's disease in vivo model. Thank you.
Thanks, Dr. Denora, for your interesting presentation. The discussion is open. Any question? I would like to know how would you like to overcome the stability problem of the prodrug?
Which one, the PCPDOP, the L-Dopa codrug? Well, actually, we are focusing now just on the dopamine codrug that were found to be stable, adequate stable in phosphate buffer and serum. So we are looking now just on those two codrugs.
Are you thinking to include in a carrier, perhaps?
Yeah, it could be a possibility in the next future, maybe.
Thank you.
A question from
yes.
Dr. Smith.
I'm thinking about what's causing the dopamine release. Is this an amphetamine-like effect that the compound is going into the vessel
Right.
and actually kicking it out? Is that what's happening, or is there an increased synthesis? What's causing the dopamine release?
Right. These Prodrugs are lipophilic. In fact, as you can see from this table, the C log B values are very high. It means that these Prodrugs are able to cross by simply diffusion the blood-brain barrier. So this is the mechanism. Once they reach the brain, they should. There is a cleavage by hydrolysis cleavage or also an enzymatical cleavage of the carbamide linker. So they can release in the brain the dopamine or the L-Dopa ethyl ester.
So the release is the release from the prodrug. It's not the release from the neuron.
Right.
Okay, I see.
Yeah. So in this case, the advantage of the, we are using this Prodrug because we want to increase the dopamine level in the brain, but at the same time, we want also to give imidazopyridine compounds like the anxiolytic agent, the zolpidem, because there are evidence that they can be beneficial in Parkinson's disease patients.
Any other question? Thanks, Dr. Denora.
Thank you.